you are very welcome to join. And it's really a pleasure to have Hannah here today. And the idea is to give you a glimpse on a very successful transition from academia to industry. And I mean, I need to read because Anna's series is impressive. Anna's initial trajectory is very similar to the one that actually our students make and made, because she got prizes during the high school. She got a prize from the German Physics Society, as I guess as a high school student still. What is it? And then she was admitted at Cambridge in a very interesting bachelor course, which is called Natural Sciences, which could even be an inspiration for us, because in the first 2 semesters, students from life sciences, chemistry, and physics sit at the same courses. And then after the first year, they diverge. So then after that, she actually worked for her Master of Science in Napoli at the TIGEM. And she got her degree from the Open University, and then she was a PhD student at Stanford. She got a PhD in neuroscience at Stanford at 2011. And then Hannah and Giuseppe, her husband and co-founder of MMI, made the very bold choice of coming back to Italy. And incredibly, this has been a very successful choice, because they founded MMI, and I think MMI now is a company with more than 100...
280 maybe.
Yeah, 280 employees, and the main building is here, is in Pisa. So it's, you know, the demonstration that even in Pisa it's possible to build, to go from startup to a successful industry. So now it's really a pleasure to have you here, and I'm really looking forward to your talk. Thank you for accepting our invitation.
Thank you. So thank you, Sandro, and thank you to the faculty for extending the invitation. Thanks to everyone for coming to listen. I saw the description of this series of colloquia, and the idea that it should really be, you know, accessible, firstly to undergraduates, and then also to, you know, individuals from lots of different faculties. So I hope that I kind of satisfy that requirement. My idea today is to tell you a little bit about my journey in the medical device industry specifically, and kind of how that links to the skill sets that you know, you acquire, basically even training in along pathways that could take you into academia and basic research. So for those of you who are, you know, I mean, obviously some people in the audience are already, you know, have made their career paths and choices and have made those very successfully, but maybe some of you are still wondering about, you know, what can I do with basic science training other than going into academia. And so I'd like to kind of, you know, give myself basically kind of as a case study of that. So as I'll start by introducing so this is kind of you can check afterwards whether I did what I was said I was going to do, because that's a basic principle in medical device. First you say what you're going to do, then you do it, and you have to be able to cross-check in a process called verification validation that you actually did what you said you were going to do. So I want to give you a couple of case studies basically on, you know, taking technology and developing it into a setting that then brings it into the clinics, and how, you know, specific skills that you can acquire during you know, during scientific training can be applicable to those fields. And a bit of a personal intro, a bit of a professional intro, and then kind of some, let's say, product stories and how it links to how it links to specifically that kind of scientific training. So I was born and grew up in Germany. I went to a school of the European Union, basically. So I mentioned this because I think it was quite influential also on me. So basically, from age 6 to age 18, every day I was together with Italian children, French children, Dutch children, German children, English children. So that was quite a kind of, it was a coincidence because in the city in which I grew up, there was a research institution of Euratom, basically. So there were these schools of the European Union. So I kind of grew up in a surrounding that was, you know, very, how can I say, very colorful, and made me a true European at heart, despite being half American. And then I went to the University of Cambridge as an undergraduate. During high school, I already started developing interest in neuroscience, and kind of especially in like language acquisition, language processing, maybe partially because of that kind of multilingual, multicultural surroundings. And then during my undergraduate, I developed an interest in developmental biology and developmental neurobiology, and then kind of stayed on that track for my master's and my PhD. But then during my PhD, because sometimes things happen that are, you know, kind of beyond your control, the financial crisis in 2008 happened, and there were, at least in the US, like there was, you know, like no research funding, like no cookies anymore at like happy hours and that type of thing, because a lot of the academic system is based on endowments, which is based on, in turn, basically the stock market. So that was like, so many really, really gifted people around me couldn't find postdocs, et cetera, et cetera. So I also kind of, that was like one shock to the system, I would say. And then at the same time, I was, you know, being at Stanford in that environment of tech transfer, of Silicon Valley, of really taking, you know, basic research into you know, startups, and also of kind of building on applied research, basically. So it was kind of a combination of factors in terms of like time and space, and then also in terms of meeting specific people, among which, as Sandro mentioned, my husband, basically. So that comes into my personal background. So my father was German, my mom's American. My father was also a medical device entrepreneur and had like a little, you know, for a number of years, a company in our basement, Medizintechnik Teichmann, where he had like a dental scaler, early ultrasound plaque removal system that he developed and commercialized, built and commercialized. So I think that was kind of also like somewhat influential. I'm the youngest of 3 sisters. And then during my PhD, I met someone whilst I was at Stanford who was an engineer at a medical device company called Intuitive Surgical. We got married in City Hall in San Francisco. That's Giuseppe. And then basically we decided to move back to Italy. We co-founded first a consulting company called MedRA, or Medical Robotics Applications. We both basically, you know, obviously in different domains and in different capacities, consulted out of that. Giuseppe hired someone who ended up being the 3rd co-founder of MMI, hired him into MedRA, and was consulting for Intuitive Surgical, and at the same time developing basically technology and evaluating those technologies and deciding, well, what could be an interesting technology to found a company out of. So I was kind of consulting in other, like on upstream product development, acting, for example, collaborating with the Stella Maris in Pisa, and a kind of design for all toy company, acting as a bridge between clinician end users and the company, basically. And then one of the core technologies that Giuseppe and Massimiliano developed was a miniaturized wristed robotic instrument. And that kind of core technology of a 3mm wristed surgical robotic instrument became kind of the launching board of then what was MMI. So we co-founded this company called Medical Microinstruments. I covered the function of what was called VP Clinical Development Medical Affairs. And I'll tell you that story kind of in more detail. I stayed at MMI until the end of 2021. And then basically the company was around 100 employees at that time. It was becoming a little bit kind of more, you know, verticalized in terms of functions. And I decided to like go back to consulting and advisory, which is what I've been doing for the last 4 4+ years. And amongst that, one of the kind of more interesting, or like, let's say maybe like more long projects that I worked on is on an implantable neurostimulator. So I'm going to tell you that story as well in terms of like a product example. And then just recently, at the beginning of this year, I've now joined a French company as chief clinical officer in Paris called Roboté, which it's a deep tech neurotech company with a micro robot for brain biopsy and local drug delivery. So that's kind of my professional development. I'll tell you a little bit about MMI and also specifically how it kind of fits, like how the basis that I covered, how that fits into, I think, a lot of the skills that you acquire basically in basic science training. So as I said before, the kind of the core technology behind MMI is a miniaturized wristed robotic instrument, which basically means that compared to, let's say, kind of leading surgical robotic systems on the market, such as the Da Vinci robot by Intuitive Surgical, which was designed and is, extremely elegant and powerful for general surgery and laparoscopic surgery, this miniaturization of the technology basically allowed us to address other clinical indications. So that was kind of, let's say, the research part that I was, the first research part that I was on and dedicated to is looking at, well, what can we do with this technology? Okay, so one of the themes basically is like the research and kind of analytical and like experimental design skills, et cetera, that you acquire from like undergrad through PhD, that also applies to researching users, and then also generating data around, in my case, devices, basically, right? But that also applies to biotech and pharma. So at MMI, we started with this miniaturized wristed robotic instrument. And the initial research that I did was basically, well, what can we do with this? What can we do with this very, specific and kind of miniaturized technology? It's what I call today high precision open surgery. Okay, more specifically, it's what is called reconstructive microsurgery. So basically surgical procedures that take place under the microscope with very, very high precision, but not in laparoscopy, basically not with keyhole surgery. And so the areas, let's say, that I kind of covered was like making that connection from the core technology to the market fit, looking at mapping different indications and mapping the clinical needs across those indications. And then also understanding, well, amongst those users, basically, and amongst all the stakeholders that there are in medical device, what is the data that we have to generate to be able to speak to those different stakeholders, right? So that, who are they? They're regulators, okay? So essentially, notified bodies and competent authorities that are responsible, for example, in the US, the FDA, in Europe, it's basically by country and notified body, that are basically responsible for giving market approval, right? Because these are highly regulated products that basically have to go through a regulatory process to be able to get to market. So regulators want certain types of data around what is called safety and efficacy. The clinician end users also want certain types of data around efficacy and kind of what are the benefits for them. Payers will want certain types of data around, how is this beneficial to healthcare systems? Patients will want to see certain types of data, et cetera, et cetera. So it's basically part of that job is like understanding who the users are and understanding what type of data they need to be able to start to adopt a technology, basically. Yeah. Montachiello, via Giannessi. Pisa, Montachiello. Do you know the Forti area, the industrial area of like Centro Direzionale, via Giannessi, it's called, the building. Yeah. Near Amazon. Do you know there's like the massive Amazon? So basically R&D, so research and development and manufacturing is in Pisa. and like general administration, marketing, sales, et cetera, is in Jacksonville in Florida for MMI today. So it went kind of through this like pathway of like, you know, the ideal. I was again kind of a combination of like, you know, serendipity and capability of this kind of ideal pathway of starting with angel funding and then raising VC funding, multiple VC rounds, and then, you know, getting through various basically regulatory milestones along the way. And this is kind of the, you know, this is the, as a background, really kind of the evolution of what one of our co-founders, Giuseppe Prisco, saw in a company called Intuitive. Joined in 2001. There were about 150 employees of a company now that today probably has like 10,000 employees, 8,000, 10,000, something like that. So it basically, you know, really from like first clinical product, going through multiple device iterations, bringing in new product lines, et cetera, et cetera. So really having lived firsthand that kind of growth trajectory of a surgical robotics company, of like a medical device powerhouse, but then also seeing that within that product offering, there were gaps that, you know, of clinical indications that could still be addressed, basically. And specifically around size and miniaturization. So that's kind of like where the initial product development that Giuseppe and Massimiliano worked on in 2013, 2014 focused. And so basically my contribution, I'd say this kind of sentence here, like I should probably tattoo that on my arm, because that's like my learnings of, you know, the last 10 years, basically. Like what basically are kind of like the building blocks of a potentially successful startup, right? You have to have proprietary technology. It's like an innovation that is really innovative, okay? And that you can somehow build a moat around. That innovation also has to correspond to a real clinical need, okay? Like there has to be a driver, there have to be people who will in some way benefit from that innovation. And then there also has to be, you know, a real substantial market opportunity of potential annual revenues that correspond to that clinical need, right? So, you know, having a really cool innovation that is, you know, for a very small market, or having, you know, something that is, you know, addresses a large market, but you can protect, et cetera. Those are all other companies. That's a different model, right? Okay, so basically I'm telling my story today. Like obviously there are other models of like entrepreneurship that can be highly successful, right? Like Antico Vinaio or something like that. That no, I'm serious. Like this is not the only way to build a company, but this is one way within medical device and within biotech where you really need these building blocks, right? You need to have something that you can protect. You need to have a real clinical need, and you have to have a real sizable market, basically. And when you put those things together, maybe you'll have success. So, and you know, this was kind of my learnings. Obviously, I'm not the first person to discover that, so it kind of corresponds like quite closely to what, you know, Tim Brown from IDEO described like in the early 2000s, the DVF framework, like something that desirability, viability, and feasibility. Somebody needs to want it, the desirability. It needs to be viable from a market standpoint, so there needs to be a substantial market behind it. And then it needs to be technically feasible, right? And now more recently, that kind of the responsibility and sustainability aspect has been added to it as well, which I think is a real, you know, very important and very challenging part, especially in medical device. So you kind of have these building blocks or pillars, and especially in the case of MMI, we really built the product around around those principles, basically, because we had a core technology. But we could have also, which is the miniaturized robotic wrist, so that could have basically also been a platform, let's say, just for eye surgery or just for hand surgery or something like that. But that would have not had a large enough market to basically be able to attract venture capital. So as I, in the early days, basically interviewed different surgeons, studied all the databases, et cetera, studied databases of like volumes of surgical interventions, that then fed into the user requirements that I, or like my group and I, developed, which meant it had to be a multi-specialty platform. Okay, this couldn't just be like an eye surgery platform, because that wouldn't be a large enough market, basically. So basically, whilst the core technology was the wristed robotic instrument, the whole platform architecture, which basically means that which I'll show you, the whole platform architecture in terms of being able to operate on many different anatomical districts. So this is the core technology of the wristed robotic instrument, and now in a minute you'll kind of see the scale. So that's a 3 millimeter diameter instrument at the wrist. That basically then fed into a whole platform architecture that makes it a multi-specialty platform, okay? So it's within like this, the field that is called medical device, right? So not biotech, not pharma, and basically it means like it's an instrument or an appliance or software or whatever that is meant for diagnosis or therapy, essentially, like in large, like at a high level. And that basically falling within the definition of what a medical device is, then that means that you have to follow a number of processes. And some of those processes basically regard also generating preclinical and clinical evidence. So what are the skills that you're acquiring, like as you go through Scuola Normale, etc. So one thing that I think is really important that I hope you're learning, and that I think is like really, kind of should stay with you, is basically the ability to ask questions, right? Like, so why is something like a certain way, right? And so like one kind of anecdote that I want to bring is like, so I asked like in my, one of my neuroscience professors, we were kind of doing like a module on like neuroplasticity, and he brought in like the stratum, the glasses that invert your vision, right? And basically, demonstrated that within like the time frame of like 10 minutes or whatever, you learn to catch a ball. You can learn to catch a ball even like with your visual field inverted, right? I said like, hey, Matthias, like how does this fit with Sperry's experiments of surgery on the frog eyeball inversion, right? Where basically the chemoaffinity like hypothesis was generated, where Sperry continued to observe the axon like migration to the same target, so not the inversion. And the frog basically continued to live with an inverted visual field and wasn't learning. And Matthias Landgraf, who's like professor at Cambridge, I decided to do my thesis with him. He was like, I don't know, like maybe it's, a difference between frogs and humans. Maybe it's a difference between age period, like that, in early stages, you have, a different kind of the time frame of development has like fundamentally different characteristics to the time frame of like adulthood, et cetera, et cetera. But I think like I wish for those of you who are students here, basically, that like you continue to like be able to ask questions, and that you also find people who are able to say like, I don't know. And here I just want to, like, in case anyone, like I keep thinking of this poor student, the medicine student from Caserta, okay? I don't know whether you guys remember that like during COVID, there were all these online exams. And there was this huge scandal of like a medicine student from Caserta who said she was asked like, oh, does mitosis continue after death? And she said yes. And there was like this massive scandal. Like the professor started like shouting at her during the exam. Her mother came in. It was like in the Corriere, Selvaggia Lucarelli, and then I was like, well, you know what? She's right. Mitosis does continue after death, basically. So I just like basically like there are all these publications, right? And to my knowledge, like they haven't been debunked, right? But basically, like sometimes things aren't intuitive, and you have to basically continue to be able to ask questions, ask why. And if you find people that think they're like the owners of truth, then that's like a massive red flag. Like that's going to be, both within academia and also within industry, that's going to be a real roadblock, basically, because you have to continue to be able to find different ways of basically, getting to an answer, getting to a solution, getting data, etc. So I wrote the Corriere letter to the editors to, like, in defense of this medicine student of Caserta that didn't get published. But anyway, I hope she got her medicine degree. So your ability to ask questions is core. Your ability to, like, think critically about stuff is core. Your ability to design experiments and kind of, you know, tease apart ways in which to get information, yeah, both about user groups, about clinical indications, but then also about the device or molecule itself. And then also your ability to gather and analyze data, again, both about the users, that's kind of like the upstream, you know, upstream marketing or product development, product management part, and then about the device and the product itself. So all of that data about clinical safety, clinical efficacy, et cetera. So, and then when you combine those, like those processes and those understandings together with the processes of design thinking, I think that that's, you know, gives you basically kind of a unique take on how to contribute and how to kind of like accompany a product through its life cycle, because products have, you know, a life cycle from like inception to you know, that early kind of development, that gathering the data, then bringing it to market, continuing to gather data, starting to think about new indications. So that's something that I'm going to talk about as well, that there are always surprises, right? As a device kind of, you know, as a device grows, starts to be used by clinicians that maybe, you know, are to the left and right of kind of that initial core indication that the device was developed for. And then also, as our just understanding of human anatomy and physiology develops, also new indications can develop. So I think that's also a really interesting, let's say, opportunity where kind of scientific abilities are valuable. Yeah, and then basically, if we kind of look at the MMI history and the product life cycle, like all of these stages, right, required research around the users and the clinical indications, generating preclinical data. So actually, Sandro supported us along the way, in 2020, 2021, on analyzing some of the data, and then generating the clinical evidence as well. So there was kind of that, you know, and that's, you know, those are efforts that continue. I kind of showed here like those 7 years that I was like closely involved in those programs, but those are really, again, like patterns that are kind of needs that don't expire, basically, like in the life of a medical device company, or of it's similar in biotech, basically. So specifically, you know, regarding the MMI story, there were these 3 founders. Giuseppe and Massimiliano developed like the core technology of the wristed robotic instrument, and I worked on basically kind of making that match towards the potential indications and then generating the evidence around that. And what came, you know, what is the product that was basically born from there is here at the end of these small rods are the wristed instruments that you saw in the video before. It's what is called a teleoperated robotic system. So essentially, the surgeon uses like what are called master instruments or joysticks to basically guide or drive those wristed robotic instruments. And the robotic instruments are the ones that are on the tissue themselves. Okay, so what is the tissue that is being, you know, typically handled by this surgical robotic system, which is called Simani? It's basically designed for suturing very small anatomy, starting from vessels, so veins and arteries, in what is called free flap reconstruction. So essentially looking at all of post-traumatic reconstructions, post-oncological reconstructions, lymphatic surgery, and now even like cervical lymphatic surgery, which could be a therapy for Alzheimer's. So why did we make that match between robotics for microsurgery rather than, you know, building a system for, I don't know, watchmaking or just, you know, hand surgery or something like that? Well, firstly, it's a clinical kind of indication or field where the success is like very much determined by the dexterity and by the precision and the quality of this gesture called anastomosis. So basically suturing 2 vessels together. that's a level of precision that is very difficult to achieve, because basically human physiological tremor is kind of at the same order of magnitude as the gesture itself that the surgeons are trying to perform. It's basically like an athletic feat, right? So you're suturing 1 millimeter diameter vessels under magnification, under a microscope. You can see the surgical microscope here in the picture on the left-hand side. And then there are also, very long procedures. So these microsurgical interventions basically are, from a surgical standpoint, extremely tiring, because the reconstruction part is right at the end. So basically, initially, they'll typically demolish or export the tumor. So for example, in the case of a breast cancer, the tumor will be removed, and then the flap will be harvested, and then the flap will be put at the host site, and then the anastomosis is carried out. So by the time they're doing this very, very small, precise suturing, they've already been in the OR for 8 to 10 hours, basically. So the robotic value proposition is really in added precision, in facilitating that very difficult kind of manual task, and then therefore improving outcomes, okay? So that's where that data generation kind of task started to, identifying that clinical need. So this is an example of that type of procedure, right? So tumor demolition, this is what is called the donor site. So this is, for example, some of these reconstructions can also be carried out with prosthesis, okay? So for example, you can have a silicone implant. But for example, in head and neck reconstruction, microsurgical reconstruction is sometimes the only therapy that is really feasible or viable, right? So in this example, this is what is called a deep epigastric perforator flap. Basically, the tissue is taken from the abdomen. This perforator or vessel basically is dissected and clipped basically at the donor site, and then brought to the host site, and that's where it's revascularized. So basically, the local host circulation is basically sewn together with the flap so that it becomes revascularized. Same thing with, for example, digital amputations, workplace accidents, et cetera, et cetera. And then finally, this is an example of what is called lymphovenous bypass or lymphatic surgery, where in cases of lymphedema, which is basically when lymphatic fluid accumulates due to malfunction of the lymphatic system, either because due to cancer, the lymph nodes have been, exported or asported. And then there's basically a local suturing of the lymphatic vessels, which are about 0.5 millimeters in diameter, to the veins, so that the lymph fluid can go back into the venous system, and the arm or lower limb or whatever isn't as heavy and bloated, basically. So these are some of the clinical indications. But as I said, for example, now in the last couple of years, new indications have evolved. So there's been a description maybe 2 to 3 years ago of what is called the lymphatic system in the brain, or basically the glial lymphatic system, and how essentially protein accumulation within the brain lymphatic system may be contributing basically to some neurodegenerative diseases. So now they're starting to do cervical lymphovenous bypass basically as a possible therapy for Alzheimer's. So that's an example of how they're, both with kind of the life cycle of a product, but then also evolution of our understanding of anatomy and physiology, new indications can be born. And here's an example of the this is a prototype on the left-hand side, right? This is not what this was in the early days during an animal study, but on the right-hand side, you see the robotic wristed instrument, and on the left-hand side, the joysticks or the master instruments. And I just go here. I want to tell you a little bit, just kind of at a high level. Here, if this I think kind of shows you quite nicely. This is when it's carried out manually. Okay, so this is a microsurgeon. This is a fully trained microsurgeon, someone who has a degree in medicine and a specialization in plastic surgery and does this as a profession, but all has amazing surgical abilities, but still has physiological tremor. So basically that's where kind of this smoother, like robotic approach we thought could bring clinical benefit. And so then how do you go about like designing a study to prove that, right? Because that's just a hypothesis that like getting rid of the tremor is going to help somehow. So then what did we do? Firstly, we looked at like, well, what are the models for this type of surgery? And this is actually in a model, right? So this is in the rat femoral artery suturing model. So basically I designed a rodent study, and in both artery and vein, we basically over like a series of 56 animals basically asked the surgeon always to suture manually on one side and robotically on the other side, okay? And then when we pooled the data, so what happens, like what is the risk from a clinical standpoint in these other than it's being difficult? The risk is like a thrombosis can form, okay? So there's basically at the point of suturing, you start to get turbulence, you start the thrombotic cascade, and a thrombosis can form, and basically the flap, that piece of autologous tissue, is no longer vascularized and goes into necrosis. So that's like a tragedy, right? For the patient who now has a wound, like on one part of his or her body. The hospital and surgeons who have been working for like a day and then have to bring the person back into the OR, etc. So the event that you really want to avoid is basically this is thrombosis, okay? So the patency rate is like the inverse of the thrombosis. You want the patency rate to be as high as possible. And what we found in this, in the animal study that we had started like prior to having the device on the market, right? Just like immediately when we first had a prototype that had all of the operating principles, okay? It had none of the medical device regulation. It wasn't done under what is called design control, anything, but we had a functional system. And we got approval from the Ministry of Health for a pilot study to start to gather data on basically a potential clinical benefit of this surgical robot. And what we found over multiple studies is that basically the robotic approach halved the thrombosis rate, okay? So you were actually like reducing, this would be the equivalent of reducing those events where the patient has to go back into the OR or where the flap is lost, basically. And yeah.
But the legal clinical service, the obstacle much better.
Yeah, okay, right, yeah. But then it's blatantly obvious, but well, I mean, how would you know already from that like there's actually a clinical correlate in something that matters, right? Because you can say like, I don't know, like you can, emotion can be done in multiple different ways, right? So the question was really like, is there like a real clinical benefit other than it's just being like prettier and smoother and nicer? Is there something that is actually, going to be helpful to patients, to clinicians, to hospitals, et cetera? And it's not a trivial question because it's still an open question in robotics, right? I don't know whether this is something that like any of you are like aware of, like when you bring a new technology into a hospital and it's very expensive, okay? These are technologies that are expensive to develop and they're expensive for the hospital to buy. And so, when the healthcare systems end up having to be quite discerning on like, what do we use this technology for, right? So for example, the Da Vinci robotic system, yeah, it has lots of, advantages in terms of like cognitive load for surgeons, et cetera, because basically the software makes it such that when you move to the right, the instrument, like when your hand moves to the right, basically you on the screen, you see the instrument moving to the right. Whereas like in classical manual laparoscopy, because you have the fulcrum basically around the trocar, the manual laparoscopic surgeons have to do all of the inversion, this cognitive inversion, right? So the Da Vinci robotic system got rid of that. But that's not necessarily a clinical benefit, right? So there, in medical device in general, but like robotics specifically, there's a big kind of, I would say, burden on the manufacturers, rightly so. I think it's right, but like rightly so to show, well, this is actually going to make a difference, basically. So we started basically as soon as we could, you know, in an animal model. We were lucky that there was a validated animal model that was already used for training. And then here, basically what we did then was basically put together, once the product was on the market, or together, let's say kind of as an agreement with a notified body, put together a post-market study to gather like large body of data on the clinical outcomes. Okay, so there's one in Europe that has, you know, Primo, this is the study that I designed and got approved. And then there's an analogous study. This has about 500 patients in it now in Europe and is closed. And there's an analogous study ongoing in the United States with a similar, let's say, like what is called registry design, where basically you're gathering real world evidence on similar types of outcome measures, right? So basically, how often do they have to convert to a manual procedure? How often do they have to take the patient back in to like revisit the flap? How often are there flap failure events, et cetera, et cetera? And the interesting thing is that, you know, what is it now? 5 years and 500 patients later, we're starting to see the same thing. So this pattern basically of decreased thrombosis rate that we saw in the early days in the animal model, that has kept true in clinical real world scenario, which is like incredibly powerful and in a way lucky, and also rare that you get basically that kind of one-to-one translation from your preclinical model to your clinical data. And what we also found is basically a histological kind of, you know, yeah, go ahead. Yeah, so we put together a surgeon training program, basically, which started from like a synthetic model, so basically silicon tubes that are used for training also of manual microsurgeons. And so we basically, you know, put together kind of a minimum number of like anastomosis that you would have to do on these like tube models, because they're already fully trained as surgeons, right? So like the company doesn't train them as surgeons, they only train them on the device, right? So basically the company uses these synthetic silicon vessels, and then also tracks basically as they go through training that they come to kind of what we would call a plateau of their learning curve, basically. That they, you know, that they've come to a time, like a time per anastomosis or per suture that is no longer basically being reduced. So it's typically about 10 anastomoses, something like that is usually like what is required. Yeah. So they basically, you know, when a hospital decides to install the system, the company also basically provides training to the surgeons and also kind of certifies that they're able to use the system safely. Yeah, and here this is like a picture of the system in the OR in Florence in one of the early clinical cases. And now I'd like to talk maybe a little bit about risk, in case there are people here who are like interested in entrepreneurship and kind of that path of like how data and collecting data and preclinical evidence and clinical evidence, how that fits into risk and de-risking a company. So basically, you know, we didn't have the means, as I said before, these are, you know, very capital intensive projects, right, or endeavors. So we knew from the beginning that this wasn't something that we could fund ourselves, right, the founders, okay? So we were aware that we were going to need investors, essentially. And so when you think really about like what is a company, right, in the early stages compared to when the device is on the market, when it's commercial, when it's clinical stage, et cetera, one way to think about it is in terms of risk, okay? So basically at the beginning, there's nearly only risk, okay? You have risk around technology, you have risk around the founder team, you have risk around the market, and the way to basically kind of be able to attract in a credible manner, you know, continuous investment is basically with each investment continuously de-risk the company, right? So basically identify at the beginning, like what are the biggest risks that we have today, and start to, invest in your activities that are going to reduce that risk, so that when you go to fundraise for our next round, you can say, okay, I've reached this milestone, we've done this and this, we can now show you that we're like this much closer basically to being a company that is, close to clinical and commercial stage. So essentially in our case, what did that mean? Like with the initial, really just kind of the prototyping and initial kind of instrument development, that was bootstrap basically. But then once we had that 3 millimeter diameter instrument without a robot, we basically went to angels, and we had already, gone to a conference, had surgeon interviews, had done like basic market research, etc. We had done all the things that were cheap, so to speak, right? And basically started to de-risk that kind of market fit question. We hadn't de-risked we had de-risked just like the core technology around the instrument. But then progressively, basically with each investment, prior to going to raise the investment, we tried to basically obtain milestones and data that could show we've done this, like we're this much closer, and basically gathering preclinical data and clinical data. That's like those are major milestones for companies of this type. Yeah, go ahead. Yeah. Since 2022, yes. So basically, like you can see here, we got 3 million from like basically alumni of Scuola Superiore Sant'Anna. Okay, so basically the Italians, like basically Italians, Europeans in the first like 3 million. Then 20 million from a syndicate of 3 European VC funds, Panaches, which is Italian, and DELLA, which is French, and Fountain Healthcare, which is Irish. And then we got a loan for 15 million from the European Investment Bank. That was repaid like very rapidly. But then the last, like these 75, that was a syndicate of 3 US VC funds. And then the Series C, the 100 million, was Fidelity, basically. No, so basically in general, I think it's a very difficult moment to raise VC funding in medtech, but just due to, like, it's kind of it's a little bit of another question, but like in general, it's become, I think, more difficult than it was when we founded this company for a number of reasons. Not due to tariffs, but basically, there are kind of being constraints. Yes, there are constraints in terms of there being, let's say, there's more capital in the United States of this kind of size, 75, 100, etc. It's pretty rare to see that type of round in Europe from European investors. Sometimes happens. Like, for example, last year, Salvia, a Dutch neuromodulation company, implantable neuromod company, raised a European round of 60 million, I think. So it happens in brain. Another implantable neuromodulation company raised a round of like 50 million, I think, with Merck and private investors. But in general, it's, let's say, there are more like of these very large tickets basically in the US, but that came in the case of MMI, and also, I would say quite frequently with the condition of redomiciliation. So basically the company was relocated, became a US company essentially. So from a legal standpoint, from the legal standpoint, basically it's a US company with operations in Italy. Like it has, our R&D and manufacturing in Italy, but the US investors obviously had, kind of the wish to have that like very large investment they were making within their own jurisdiction. So I wouldn't relate it necessarily to like, this like current administration, but just to kind of, the dynamics of like what expectations of investors are when they make that size of investment, basically. Yeah.
Back to my the lack of environment that
I don't know what you mean with like in principle. I mean, there are like rounds of this type maybe in like, you know, in other like industries, like, you know, Mistral or like in AI, LLMs, etc. But like this type of ticket, you know, for medtech from European investors for medtech in Europe is like pretty rare. I mean, I can like, as I said before, there, like when it happens, I can name it like Salvia or, you know, like
the approach of the investor, like
oh yeah. Okay. So it's a cultural thing. Yes. So basically, in the United States, so pension funds invest as LPs in VC funds. So there's like a whole kind of how can I say? There's a whole like market of like asset allocation that is distributed differently in the United States than Europe. Yeah. So no, there are underlying dynamics that are different in the United States and Europe. Yeah, and then this was mentioned in 2020, at the end of 2024, as one of the best inventions. The company's grown significantly. We're getting to the end of the hour, and I want to kind of loop to another example within which I think, you know, from having done neuroscience as an undergraduate and also you know, having the connection to Sandro and maybe some of the people in the audience, there's like a huge boom at the moment in what is called neurotech, partially driven by BCI. But this is kind of, at the moment, there's like this second wave, basically, of neuromodulation, both invasive implantable and non-implantable neuromodulation. And that was a kind of an area that I've worked in quite a lot in the last couple of years, and that I think is also, you know, super interesting, and where people like yourselves who have like a basic science understanding can really find applications in industry and lots of like interesting companies, basically. So the basic kind of like the biggest market in neurostimulation and neuromodulation continues to be pain or like spinal cord stimulation. But there is this like new wave basically of neuromodulation, neurostimulation, and also BCI or brain-computer interface, basically, which has started to really take off in the last like maybe 5 to 10 years. And one company that I worked with, which by the way is an area that is continuing to attract, like within medical device, continuing to attract quite a lot of funding. So within medical device, within the last couple of years, one project that I worked on was basically developing a new indication for a device that was already on the market for epilepsy. And this is a German company that has a subgaleal implant. So basically it doesn't require a craniotomy, but the device is, again, on the market for drug-resistant focal epilepsy. And this is kind of the operating principle. So rather than requiring a craniotomy, the way like many implantable neurostimulators do, this is a pseudo-Laplacian electrode with a center and like 4 surround electrodes. It is basically, you know, screwed onto the cranium. So the periosteum is cleaned, and then this electrode array is attached, and then there's an implantable pulse generator, like similar technology to pacemakers, that is implanted under the clavicle, basically. And this has like very, very positive results for, you know, very good therapeutic benefit for drug-resistant epilepsy. And the company came to me and said, hey, we want to do kind of like a startup within our startup. Can you help us identify, like do the research around what are indications where we could use this technology, changing the stimulation parameters and changing maybe where we implant it. And then basically, you know, so help us find out where clinically, what market can we go into and bring patient benefit, and it's a sizable market, right? And what are the device parameters that we would have to redesign there, but only touching software, please, okay? So don't touch any of the hardware. That was don't touch any of the external parts, because then all of the biocompatibility, form factor, et cetera, would be lost. So we want to do something lean from like a time frame standpoint. So I did, this is basically, the data that the company had already generated around epilepsy. So they have a very good responder rate. So decrease in number of seizures that these patients have in drug-resistant epilepsy. And then based on that, we basically looked at, this is speaking about the analysis for payers, right? So I also did an analysis on like, how do healthcare systems save from reducing, for example, seizures, right? Because that's also an important part of the analysis in this type of picture. And what we basically identified was looking into, well, maybe stroke rehabilitation, maybe treatment-resistant depression. And we identified, so this is basically looking at this kind of minimally invasive, like miniaturized technology compared to the more invasive technologies that are on the market for epilepsy, which are either deep brain, like needle electrodes, or closed loop systems, but they're extremely invasive, or not so invasive but not so effective, basically vagal nerve stimulation systems. So this technology is really like in a space where it's highly effective and not very invasive. So we want to leverage that kind of uniqueness, and I identified treatment-resistant depression as a new indication, basically. And so we started to work around that together with psychiatrists in Germany to identify basically, a new product introduction project for treatment-resistant depression. So basically, device redesign in terms of new stimulation parameters, looking into clinical literature and understanding, well, what is working today for this patient population in terms of brain stimulation? How does that translate onto this implantable system? And then what is a kind of lean clinical study that we could develop to generate the data for the efficacy of this new device? So I'll just get to kind of, one, for example, the transcranial direct current stimulation is a non-invasive technique that, doesn't have very effective, like very kind of positive data towards it, or very like persuasive data in large trials. So we decided not to kind of go down that route, but we did go down the route of the stimulation parameters used in repetitive transcranial magnetic stimulation. So these are like systems with like large coils basically that create a magnetic field that induce, basically evoke potentials in the patient. So it's, again, not invasive, but there are a number of different kind of stimulation parameters that can be applied to these coils. So basically I went into literature and looked at, well, what stimulation parameter in terms of hertz, in terms of, basically in terms of frequency, in terms of number of sessions throughout the day, et cetera, would be appropriate to translate onto an implantable device. And basically what we came up with there is basically this, based on a study from a group in Canada, this 3D trial, which had, very good outcomes for this, what is called intermittent theta burst stimulation. Okay, so they didn't invent intermittent theta burst stimulation. I didn't invent intermittent theta burst stimulation, but it has very good clinical outcomes for treatment-resistant depression patients. And so basically, we kind of worked with this. This has all been shown at other conferences. Like everything that I'm showing you today has previously been disclosed, right? So we worked around a device redesign based on intermittent theta burst stimulation, and developed a clinical study, basically changing both the, let's say, the polarity of the stimulation from inhibitory for epilepsy patients to excitatory for the treatment-resistant depression patients, identifying, a new personalized implantation location, and then putting together a double-armed sham-controlled study in 3 centers in Germany for the first in human, and taking this through ethics approvals, competent authority approvals, et cetera. And that was a very, I would say, a very nice device, a nice kind of chapter of mine of the last couple of years that I think is really within a field that I think is, I can highly recommend to people who have like backgrounds in neuroscience or interested in entrepreneurship or interested in industry, I think like the neuromodulation and neurotech space is like really interesting and taking off at the moment. And there's a lot of space for people with kind of basic science competencies. And now, basically since last week, I've started on a new adventure for a company that has this micro robot that you can see on the hand here, that is basically kind of bringing together my experience in robotics, in neuroscience, in neurotech, and hoping to deliver a minimally invasive device for brain biopsy and localized drug delivery in glioblastoma. So I'm looking forward to exploring this new adventure, and I hope that it's been kind of it's been an interesting and like an glimpse into what we can do with kind of our basic research and academic training, translating those skills into basically skills and kind of real competencies that are needed in medtech and industry. And if any of you are interested in having a chat or like interested in internships or something like that, you can write me or WhatsApp me. Thank you. And I'm open to questions, obviously.
Lot, Hannah. I hope this will be of inspiration also for some of you. And now we are open for questions. So are there any questions from the audience?
Can you comment on how difficult it was to get the funding at the different stages?
Yeah.
And the timing of the funding, was it critical? I mean, you had to wait for them, and this put some constraint on your development?
Yeah. So I think we were in like a unique kind of position, I would say, like when we founded the company, because again, it was like a time where the stock market wasn't performing amazingly. So I think that private individuals were more like available and like predisposed to making that type of investment. So I think when we founded in 2015, we got that private investment firstly because it was like a good alternative to maybe other investments that were available to people at that time. Then also because we had like this kind of group of like real angels, basically, I can only say, because Giuseppe had gone to the Scuola Superiore Sant'Anna and he came back and there were people there that remembered him and that really wanted to support him and that were really like, a group of people who wanted to support us and did support us, both like with investment and with moral support and expertise, et cetera, et cetera. So that was kind of the first, like that first tranche really just to get us going. Then, so I think that a lot of the time it's like a combination of, like expertise and merit and luck, right? So I think that that in life that happens a lot. Like, if you don't have the competencies and the skills and stuff, like hopefully just luck isn't enough, because that would be very extremely discouraging. But often like just the merit, like maybe isn't enough either. Sometimes there have to be like a multiple like factors put together that like allow you to kind of execute. So then when it came to the VC funding, we met someone at a like European kind of robotics innovation event who had been a VC himself and like had left his fund. And he said like, hey, I want to help you. I want to help you find like a VC investment, and he did. Basically, I mean, we paid him, but like it was really, really valuable to have someone who believed in like the potential and the vision. And it was the same thing with the angels, like Franco Mosca. I don't know whether you remember, like the surgeon, professor. When we came back from the US, he said like, hey, Giuseppe, I want to help you. And like, he really did help us like kind of galvanize people. Like when someone believes in your vision, and like what you want to create and what you want to build, like that's just, you know, that's invaluable basically. So yes, it was difficult to fundraise, but we had kind of, you know, the capabilities, right? So like Giuseppe was extremely credible as having been a manager like of developing new products in like one of the biggest surgical robotics companies. So that was like, I was credible due to like my academic track record. You know, so and so kind of a combination of like, you know, merit and also just basically circumstance. And then what is maybe the most important thing is that we executed every single time. So when we got funding, we went and we did what we said we were going to do. Because what doesn't work is like taking money and not doing anything, because that's like a sure way to not get further investment, basically, okay? So you can't just like take people's money and then be like, see ya, okay? So you basically take money, I mean, because it happens, it exists, and it's extremely damaging for other founders. Like when founders don't execute, it's extremely damaging for like the whole kind of community of like entrepreneurs, but unfortunately it happens. So basically, like we took investment and then we said like, we're going to do this and this with this investment, and we did it. And we did it like repeatedly, basically. So I think that's like, that's basically kind of like a combination of factors. And like, is it hard to fundraise? Yes, it's harder to fundraise now than it was 10 years ago. It's impossible to fundraise if you don't execute. Like, you can't take 5 million and like not do anything with it and then think that you're going to raise another 10 million. So basically there's kind of a combination of, I mean, it's a combination of lots of different factors. And did we ever have to kind of like slow down? No, not really. I mean, like if there were kind of, we always basically had, because we executed every time, if there was ever like a gap, our like existing investors basically bridged it. Yeah.
Question? Oops, sorry.
I think there was a question. Did you have a question?
Yeah. You on? Bye.
So repetitive transcranial magnetic stimulation, what is that like? How does RTMS work? So RTMS isn't used in epilepsy. RTMS, this repetitive transcranial magnetic stimulation, is used in depression, in OCD, I think maybe in like essential tremor, in addiction a lot. So basically like in, you know, cocaine addicts, smoking addicts. So basically what is the principle of action is that like a magnetic field is induced, okay, around the cortex. And that basically, like if you remember from high school, that it basically induces currents and it induces evoked potentials in the neurons. So you're basically inducing, so basically the coil is applied depending on where you want to stimulate. So basically like in depression, the coil is applied like over the left, like the dorsolateral prefrontal cortex, okay? And then, you know, in OCD, it's applied like over a different area of the brain, etc. So in all of these like different neurostimulation parameters, like where you stimulate and the specific parameters that like the specific kind of frequencies, etc., of the bursts, those are like extremely influential, like from a therapeutic standpoint. And also like the companies that make these RTMS devices, they sell different coils, like different coil shapes for different indications. So, but these are devices, so like what I was working on, this implantable device, is basically like to offer an alternative to that. Because what happens with these depression patients who do like RTMS is that they go to the clinic And like one of the more, let's say, effective therapies for RTMS requires you to have like a session every hour. So they spend like 5 days at the clinic with someone every hour, like holding a coil over them for like 10 minutes. So it's, you know, very kind of cumbersome and burdensome for the clinic personnel. So for the healthcare systems, it's like invasive. You basically have to take vacation to get your RTMS treatment because you have to be there like all day, every day for 5, like if you work, like for 5 days. So basically the idea of the implantable device was like to offer a mobile version of that, right? To offer a kind of stimulation, like parameters and a stimulation therapy that, you know, is like that you just carry with you all the time. It's not visible. It's like under the skin. Sorry? Well, there are unfortunately in like both in epilepsy and in depression and in bipolar disorder, there's always like a fraction of the population that does not respond to drugs, basically. So basically you get defined as like what is called difficult to treat or treatment resistant when you have not responded to 2 or more different active ingredients given like at a sufficient dose for a sufficient time frame. So obviously if you suffer from depression or also from like epilepsy, you know, but let's give the example of depression. Someone who suffers from depression, the first line of treatment is like psychotherapy, then selective serotonin reuptake inhibitors, and then like third lines, you know, like the SNRIs, then there are kind of alternative. And then after that, basically, there's no guidelines. Basically, then there's esketamine, transcranial direct current stimulation, electroconvulsive therapy, et cetera, et cetera. So it kind of becomes like, and RTMS is like within that field. So yes, you're right. Like to date, pharmacotherapy is the first line of treatment, both for epilepsy and for depression. It doesn't work for everyone. There's usually about a 30% population that doesn't respond to 2 or more treatments. And then there's also, like for both epilepsy and like psychiatry, there's, I think, like this, you know, forgotten population, which is women, basically, which is like at some point, if you want to like become pregnant or you are breastfeeding, like usually it's recommended that you don't take your medication during that time. So I think that especially like neuromodulation is really interesting also to kind of start to offer therapies that can be basically continuous because, you know, I mean, there are a lot of people who, you know, take, you know, pharmacotherapy of some type, but then like if you have more than one child, that ends up being a long period of your life where you're supposed to not take medication, which is like, you know, a recipe for disaster, basically. So yeah.
Other questions? I have a quick one, more a comment than a question to you. I mean, for you to comment on this. I mean, you just briefly mentioned preclinical trials in animals on one hand. And on the other hand, I mean, what you presented us, these are solutions that exist today and that improve life of patients. This is not in the future. This is happening now.
Yeah.
So what is, and on the other hand, I mean, we have a very, very negative public opinion about animal experiments. So what I'm asking you is, what is the role of animal experiments in developing medical devices? I mean, would it be thinkable to still be able to develop medical devices if tomorrow animal experiments get banned?
Yeah, that's a good question. So in all of these, like in the regulations that, so a really interesting thing I would say about medical device regulation, both in Europe and you know, in the United States, is that it doesn't spell out for you what you have to do. It's not spelled out for you that you have to do a rat femoral artery study to bring a microsurgical robot to market. No, you have to bring data that, you know, prove the safety and efficacy of the device. So if you can do that exclusively in synthetic models, okay, If you can do that only in synthetic models, then that's great. And I tried to do as much like in synthetic models as we could, right? Like, so we did evaluation of like the accuracy of the suturing and stuff with the measurements and blind scoring using the radar scales that exist for microsurgical training, etc. We did everything that we could in synthetic models. Then there are some things that you today, you cannot do in synthetic models, right? So basically looking at like the, you know, the thrombosis, incidence of thrombosis and like which depends on like the thrombotic cascade, right? Or that's, I mean, we don't have like simulation systems. There are like starting to be developed some microsurgical like as an example, microsurgical training systems. There's one system called like My Anastomosis, which is like a little kind of simulator, which supposedly should also simulate whether your anastomosis would be patent or not, right? But the answer is, like to date, I don't think it's thinkable to bring a medical device to market without preclinical data, without animal studies. Like there's just like no notified body or FDA would like accept a submission to get permission to do clinical studies, right? Before going to market, you typically have to do clinical studies. Nobody would allow you to do a clinical study based only on synthetic models, okay? So one of the reasons is, for example, like you also need to do kind of like gross necropsy, right? So we were asked to look at, well, when you're, the rats that had the manual anastomosis versus the rats that had the robotic anastomosis. And it's going to be the same thing now at Roboté with the biopsy systems, right? Like a robotic biopsy versus a manual biopsy. Do that, and then let me know like a month from now whether your animal has like, I don't know, any pulmonary embolism, any stroke, any et cetera, et cetera. So there are like systemic things that regulators want to be analyzed that you can't do basically in synthetic models. So, I mean, I think it's like at the moment it's necessary. Maybe if like synthetic models continue to evolve, but the thing is that like you're really looking at safety and efficacy, especially safety at a systemic level. And like physiology is so complex that like you would have to basically be able to like simulate a whole like biological system. So like I don't see that happening anytime soon. And like to be honest, like I mean I mean the animals, like let's talk about industrial like meat farming and the way animals are treated at the abattoirs and not the way animals are treated in animal studies, because like I feel like it's a necessary evil, but they're really like, you know, the protocols that we have around like looking at, you know, how much do the rats drink, how much do the rats eat, like are they defecating, they get like a warm blanket during surgery, et cetera, et cetera. Like so basically the levels of like animal kind of welfare are really high, and I think it's good, and it's good to be that way, but I think that like there's a mismatch in public opinion around like animal housing and welfare for like the food industry compared to like the medical device and pharma industry. So I think that, I mean, that's kind of a different chapter. Unfortunately, like I don't think that there are ways to, you know, kind of circumvent that. But I also haven't really, like, I haven't seen anything, you know, in terms of housing and animal welfare, et cetera, that I would, like, you know, that I feel was not, like, well done or justified or taking into account, like, the animal welfare, et cetera. Whereas I think that, like, there's like even like the milk and meat industry and stuff, that's like I mean, that's my personal opinion, right? But that's like, if we want to like start to work on animal welfare, I think we need to start like understanding like benefit. What is the benefit that we're getting out of this compared to like what is the cost for like the animal well-being, basically. So yeah, but I don't know what, like, I don't really know what what can be done about like the public opinion kind of question. Like, yeah, and I guess that it's hard to like do kind of, to advocate for animal studies as like, you know, that's really, really difficult, I think. But yeah.